At its heart, expression refers to a gene being kind of used, but often what people really care about is not the gene being used, but a protein being made, and the protein actually sticking around to do its job. And so we have different techniques that we can use in order to assess the usage of genes, RNAs, and proteins. And so here's just a quick guide to when you might want to use what sort of technique, starting by backing up and thinking about the gene to protein relationship. Genes have the DNA instructions for making a protein, and then the instructions for making that protein are actually first copied in a process called transcription. They're copied into a messenger RNA. But first, they're actually copied into pre-messenger RNA, which gets processed through things like splicing to form mature messenger RNA. And then those messenger RNA copies are used by ribosomes in the process of translation to make a protein. So we have all these various steps along the way from going from the gene to the protein, and then after the protein's made, then it can also be degraded. So if what we really care about is the amount of protein present, then just measuring the amount that the gene gets used or expressed isn't really going to be the information that we're going after. And so scientists have different techniques that we can use to assess expression, as well as things like what is actually present right now in cells and in other mixtures at various times. And so these are just some of the techniques that scientists can use to get a look at different steps along the process. So if we think back to that classical expression of a gene, so again, often we're thinking about being expressed to make pre-messenger RNA, but there are also other genes which go on to make functional RNAs, so RNAs that act as things like microRNAs or long non-coding RNAs, ribosomal RNAs, types of RNAs that don't actually go to make a protein. And so we can look, however, and see which genes are being expressed by looking and seeing where the RNA polymerase, which does the transcription from DNA to RNA, is actually sitting on the DNA. So there's a method called ChIP-seq where we have chromatin immunoprecipitation. And so basically you have chromatin is the form of the DNA in which it's wound around histone proteins. What you can do is you can take that RNA polymerase and you can actually cross-link it or kind of chemically glue it onto the DNA. And then you can go and you can isolate the RNA polymerase. And using antibodies that bind to the RNA polymerase, and you can kind of pull it out, and then you can sequence the region that the RNA polymerase was sitting on. And there you can see which regions of the genome were actually being kind of used by the RNA polymerase. So that'll tell you about expression at the level of the DNA. But you only have 2 copies of that gene, and you can make a lot of copies of the messenger RNA. So just knowing that the RNA polymerase is there right at this moment isn't really going to tell you about kind of how many messenger RNA copies you have or how many RNA copies you have. If you want to look at the RNA, there are a bunch of different strategies that you can use. And in part, this depends on whether you're looking for a specific RNA or whether you just want a more global perspective, as well as if you're looking for, specifically, you're looking for the messenger RNAs versus you're looking for all types of RNA. So for the most global perspective, you would do total RNA sequencing or RNA-seq. Here you take all of the RNA that's present, and typically you're going to convert that into DNA, which is more stable. You can make copies of it, and then you're going to sequence that in order to see what's present. Now, a lot of what's present, if you're caring about the messenger RNAs, isn't going to be messenger RNAs actually. It's going to be things like the ribosomal RNA, as well as those non-coding RNAs. So sometimes what people do is they actually do transcriptome sequencing. They isolate the messenger RNA, so you can do this with things like using poly-A adapters that are kind of like going to pull out the messenger RNA or enrich the messenger RNAs. And that way you're only going to see the messenger RNAs, you're not going to see all those other RNAs. More specifically, you're typically only going to see the kind of processed messenger RNAs, so you're going to lose information about things like wrongly processed products, as well as those non-coding RNAs. But doing this kind of transcriptome level sequencing is going to be cheaper. You're going to have less reads that you have to then go sift through and this sort of thing. But you're still going to get all of the messenger RNA. I say all, but really there can be some bias in the methods that are used that unintentionally enrich for certain messenger RNAs over others. So those are always things that you got to keep in mind. So even if you use that RNA sequencing and you find like a hit or you find some theoretical lead, you're then going to want to follow up with a more targeted approach. And if you're looking specifically for a specific messenger RNA in the first place, so you know you want to look for this thing, then you're typically going to go and use something like qPCR, so quantitative PCR. And typically what you're actually using is RT-qPCR. So with the RT standing for reverse transcription. So if we have RNA and we want to make copies of it, typically what we first do is convert it into DNA. So if we went from DNA to RNA, that's transcription. So if we go from RNA to DNA, that's reverse transcription. And so we can use reverse transcription in order to go from RNA to DNA. And then we can use PCR, so polymerase chain reaction, in order to make lots of copies of those DNA copies of the RNA. So we care about the RNA, so we make copies of it in DNA. So we can make copies of the DNA, and then we measure the copies of the DNA in order to measure how many copies of the RNA there were. And so how this works is that the more RNA you start with, the more copies of DNA you'll end up with quicker. So in PCR, it's run in these series of cycles. And each cycle, you're going to double the amount of the copies that you have. And you're making copies of a specific region that corresponds to the specific messenger RNA or whatever you want to look for that you want to look for. And so say you have this messenger RNA and you know the sequence here, you can have primers that kind of bind to that sequence or on either side of the sequence, involving the sequence, and then you kind of make copies of the region in between those primers, so the short pieces of DNA. And so you keep making copies of this region, and each cycle you're doubling the amount of copies that get made. If you have a way to look at the copies as they're being made, well then you can get an idea of how many copies you started with, because the more copies you start with, the quicker you're going to have a ton of copies of the DNA. And so we can look at the copies as they get made using things like fluorescent dyes as well as probe reporters that'll kind of fluoresce in the presence of the double-stranded DNA or in the presence of a specific piece of DNA. And so you have these different methods, but basically every cycle you're going to double the amount of DNA, and so you would double the amount of fluorescence that you see. And so you have some sort of threshold, and then once you get over that threshold, then you're like, okay, we are now over the threshold, and then you see how many cycles it took to get over that threshold. And that's going to tell you about how many copies were present originally. The key distinction between this qPCR and the RNA sequencing is that with the qPCR, you're looking for a specific RNA. So it's lower throughput. You're not doing a bunch of things at once. You're not testing a bunch of mRNAs at once, but it's going to be more sensitive and more accurate than if you were doing that total RNA sequencing. But both are telling you complementary pieces of information. So with the RNA sequencing, you're kind of getting the global perspective of how does this compare to other RNAs that are present. Whereas with the qPCR, you're saying, okay, well now let's get more specific about how many copies of this there are and make sure that it really is a valid kind of measurement that we're getting from that total RNA sequencing or from that transcriptome sequencing. One problem with the qRT-PCR, however, is if you care about things like alternative splicing, you're typically going to lose out on the information with that qRT-PCR because you're not making copies of the entire thing. You're just making copies of a specific region, and you're not actually looking at the copies that get made in the sense that you're not seeing the length of the copies, and so you lose information about the splicing and things, which you could get through things like targeted RNA sequencing or things like a northern blot. With a northern blot, here you run the RNA samples through polyacrylamide gel electrophoresis, transfer those onto a membrane, then use short labeled pieces of DNA, these probes, in order to go and look where those probes bind, and then you can get information about kind of the size of the pieces. And by designing the probes so that they cover different splice points, you can get information about things like the splicing. You can also design your probes for the qRT-PCR in a way that's going to give you information about the splicing. But with that qRT-PCR, with the northern blot, here you're going in and you're looking for one specific RNA, whereas with RNA sequencing, you're just like, show me what's there. Another technique that's not used as much these days, but it's kind of middle throughput, is a microarray. And so with a microarray, it's kind of like having a northern blot without the gel electrophoresis and in parallel on a plate. So let me back up a second. What you have is you have a plate, so you have a bunch of different wells basically, And so each of those wells is going to have a different probe attached to it that is going to bind to a specific messenger RNA. And then you kind of wash the messenger RNAs over the plate and then see which ones bind. And then you get a readout that's telling you which messenger RNAs are present. And so that would be a microarray. And people could make custom microarrays depending on what genes they wanted to look for. So it's kind of like a qPCR or a northern blot, but in a more high throughput manner. So you're testing multiple things at once. Again, you're losing some of the specificity and accuracy that you would get if you were just targeting that one specific thing. With any of these techniques, you're going to want to follow up and make sure you've got negative controls and things so that you're not getting these false hits, you're not getting false positives, and that you're not getting false negatives, so that if the thing is present, you're going to get a signal, but if the thing is not present, you're not going to get a signal. So we went from ChIP-seq, where we're looking at kind of usage of the gene by the RNA polymerase, to now we're looking at the RNA that's actually present as a result of that action of the RNA polymerase. Now we want to go and look at the usage of that RNA. And so again, if we're thinking in the sense of a protein, we're going to be wanting to think about the ribosomes using that messenger RNA in order to make protein. In order to do that, the ribosomes have to actually bind to that RNA and actually go along it. So there are a couple of different techniques you might hear about. So ribosome profiling, also known as ribosome sequencing, and polysome profiling. Polysome profiling, it kind of tells you how many ribosomes are on a messenger RNA, either a specific messenger RNA or in general, like how many ribosomes are there per messenger RNA on average. So there are a lot of ribosomes on these RNAs, or not very many. That's going to tell you about kind of how much the gene is going to be used kind of overall, and the overall level of the translation. More often, however, you're going to be using this ribosome sequencing, ribosome profiling. Here you're going to isolate the ribosomes where they're attached to the RNAs, and then you can sequence the sequences that the ribosomes are sitting on in order to see which messenger RNAs are being used and how much they were being used. So you can kind of get that information about the ribosomes, where they were on the messenger RNA, how many there were on each copy of the messenger RNA, because if you've got more ribosomes, that often is going to be meaning that that RNA is being used more. But if you see that the ribosomes are all kind of in a single spot, then that might indicate that you have some sort of buildup or translational block. And so ribosome profiling has a lot of different uses, and it's really cool, but it's telling you about the usage of the messenger RNAs by the ribosomes. And so we go from ChIP-seq, we're looking at the DNA and the RNA polymerase. Then we go and we look at how many RNAs there are using things like qRT-PCR, RNA sequencing, northern blot, microarrays. To go and look at translation, now we're looking at ribosome profiling, ribosome sequencing, polysome profiling. And so this is going to be telling us about kind of the usage of the messenger RNA. But now we want to say, okay, well, even if the messenger RNA is being used, are the proteins sticking around? And to go get at that information, now we're going to want to look directly at the protein. To look at the protein, here again we kind of have these multiple options depending if we want to look at all proteins versus if we just want to look at a specific protein. And so if we want to look at all proteins, then we can use mass spectrometry, which basically takes all the proteins, the proteome, and it cuts it up using an enzyme like trypsin. And so it's going to cut these proteins into these little pieces and then measure the mass to charge ratio of those pieces in order to then compare those pieces to a database to figure out what proteins were present and how many copies of each protein were present. If you want to look for a specific protein, here what you're often going to do is a Western blot. With a Western blot, here you're going to take all the proteins that were present, instead of cutting them up, you're going to run them through a gel electrophoresis, transfer them to a membrane, and then use antibodies to probe the membrane to see if specific proteins were there. So these antibodies are these little proteins that are going to recognize specific other proteins, and so you can use these labeled antibodies in order to see if specific proteins were present in the mixture and how much protein. And so that would be more akin to your qRT-PCR or your northern blot, whereas the mass spectrometry would be more akin to your total RNA sequencing. But in both of these cases, you're looking for specific proteins. And even if you were doing that kind of more global mass spectrometry, you would often then follow up with the Western blot or something like this if you were looking for a specific protein that maybe you then identified through that broader screen. Now you want to go and follow up and make sure that was real and a legit hit. Because when you're doing things in these massively parallel manners, these high throughput manners, you're testing a lot of things at once, you have to be really careful that you're not getting these kind of false positives, because by statistical chance and things, it's likely that you are going to get some of them. And so then you need to go and follow up and make sure those hits were legit.